Place. Yeah. After lurking outside of my class.
So where is the camera for the?
Have you made it to your?
It's here
Okay.
where I'm gonna stand. Yeah, I
Okay.
just was like from here. So that's the best I can do.
Chrissy, do you want to manage online questions or do you want me to do that?
I had Alex in charge of doing that.
Perfect.
So, yeah.
Even better.
But,
Yeah.
yeah.
Christie, is the Courier font intentionally signal computer use?
Yeah.
Good, okay, check. Ever seen it first?
Yeah.
I remember as a second or third year grad student emailing David Philbein to get a copy of David Cameron's thesis, and David Philbein emailed me both volumes, which I then photocopied.
Hey guys. Thanks for coming. No one's good.
Chris?
No, one of the labs.
Oh,
Or I guess people lab sessions.
yeah, somewhere I have all this information, but have you ever done any archaeological style excavation?
No,
You get the sediment and I sieve it through, as the years go by, my sieves get bigger and bigger, more things fall through.
I'm feeling that right now.
Good news is for you as well. For me, I'm just.
Hi.
It's a warmer desk developed jacket
Oh,
for this
very nice.
presentation.
Thank you.
Special paper have to clean up for you. Not for the special paper.
Oh, my goodness. Yeah. How does it work?
Oh, go ahead. Welcome, everyone, all of those who are here in person and those of you who are joining us online. It gives me great pleasure to welcome you all today to the public portion of the dissertation defense of Christy Krauss. Christy came to me a few years ago after she had been taking some of my classes as an undergraduate here at the U, and asked me, is there anything that I can do with an interest in computer science and anthropology? She finished her degree in anthropology, a minor in computer science. Is there anything I can do with that? And so we talked about some possibilities. And she started working on a model. We talked about agent-based modeling and some things to do. And it eventually became clear that Christy had a vision for what agent-based modeling could do that had come out of her thoughts when taking primate ecology and thinking that all of these hypotheses that are so difficult to test in real life with data could be tested using agent-based models in computers. And she also had some ideas about the way people were currently approaching modeling that she thought might not be the best way of doing things. And so she developed a vision for how to do things the right way. And when she joined our graduate program and started thinking about ideas, or started talking with me about what to do, she articulated some of her plans for a grand model of all of primate socioecology, that you could model all these different species. And I thought, yeah, that sounds great, but boy, that's ambitious. Maybe that might be a career goal. Well, she stuck with it and she developed that model, and she will be presenting her results and process of developing that model to you today. And this involved an extremely ambitious effort with lots of field work at the Minnesota Supercomputing Institute and using lots of computational power to create these virtual worlds. And I'm excited about what she's done and what she's found. I hope you will enjoy listening to her. Without further ado, Christy Krauss.
I feel like you did half my work for me. So I am here to talk about my model, Get. And a lot of the work that Mike has already touched on and centered on Get has to do with this question on why we live in groups, why humans live in groups, why various other primates live in groups. Primates live in groups or individuals live in groups for a number of reasons. Some benefits include protection from predators, protection from strangers, access to food resources, help from kin, access to mates. But there are also some costs, including competition for that food, competition for those mates, and spread of disease. And in weighing the costs and benefits of these different factors, many primatologists have come up with various models to explain the variation in sociality and social structures that we see among different primate species. And they include factors such as predation pressure, food availability, and the distribution of that food. And then there's some idea for how the social system emerges from this framework. And this is all very well and good, but it's all looking at kind of this macro level, looking at, population size and the populations at that level, rather than considering at the individual level, or perhaps even more importantly, at the gene level. And that's where microscale modeling comes in. So I've taken to calling models like agent-based models or individual-based models microscale models, like my advisor Clarence does. So I'll be referring to them as microscale models, but I'm just referring to agent-based or individual-based models. And there are many benefits to using microscale models. They're object-oriented. So you can directly match virtual objects that match the real world. Those objects can be heterogeneous. There can be randomness in these models. They can include multivariate capabilities such that you can include all sorts of key factors thought important in primate socioecology. It captures emergent patterns. So rather than just looking at the population size, you can actually model the individuals in that population and see what population size they have. It's accurately observant, so you can know the state of every individual in the model at every given time step. And you can use these models to explore many hypothetical situations, things that you couldn't explore in the real world, and it allows for very controlled experimentation. So I just wanted to briefly show my very first microscale model, which I did for a computer science class, which looks like this. It has a bunch of different agents in it. So some workers who go to the bank and get money and then buy food with that money. Thieves, I'm gonna just start it going so you can see it going while I talk. Thieves who steal that money from the workers to get food, and police who arrest these thieves. And then for the extra credit portion of this assignment, we were asked to include the zombie agent. And we got to play around and do whatever we wanted with the zombies. So I had fun with it and I made them kind of move more slowly across the screen like they were staggering around. And I had the other agents try to avoid the zombies. And but inevitably, the population gets too large and the zombies take over the world. That background in zombie modeling wasn't quite sufficient to model primate socioecology. And so I also conducted an extensive literature review, where I looked at microscale models in behavioral ecology to better understand how the model handles behavior. This resulted in 773 studies. And here's just kind of an overview of these studies showing by year how many studies there were. So as you can see, around the 1990s, things really started taking off. And now we have a whole bunch of microscale models today. And I looked at a few different criteria. One is, what terminology did they use? I'm forever curious about this agent-based versus individual-based terminology used. Currently, it stands we're at about 50-50, about 50-50 for who uses the term agent-based and who uses the term individual-based. I also wanted to figure out if these studies were focused on a particular species or they were just generally focused on non-specific general animal behavior. What research questions that they were focused on? How was the model reported in the scientific study? Was there any model validation performed? And did they include the source code? And from what I found, microscale models typically are highly specialized. So they are highly specialized to a single genus or species. Here's a summary of all the genera and species from my literature review, shown as a phylogeny here, highlighting that there are a lot of mammals, birds, fish, and insects in this literature review. Typically, what you see is things that you need to either model their social behavior, some spatial relationships or spatial interactions, or you need to have variation in some attribute in the population, such as variation in body size. They're also highly specialized to a single area of research. So you'll see a model on predation or a model on sexual selection. I also found that they're not realistic in some biological constraints. For example, many of these microscale models include aspatial interactions, but we know from ecology that interactions happen locally. Additionally, in some of these models, the rules for birth and death do not depend on individual characteristics, but instead depend on some carrying capacity in the population or some sex, age, class. And finally, many of these models are not, or many of these studies are not transparent in how the model is presented to the scientific community. And I want to go into that a little bit more because model transparency is very important. One needs to report the model in order for it to be scrutinized by the scientific community and also replicated. And so I focused on 3 areas that I think are really important for model transparency. One is including what is called the ODD protocol, or the Overview, Design Concepts, and Details protocol, put forth by Grimm et al. In 2006, which is a standard way of recording microscale models. Additionally, I looked at if the study described model validation. Model validation being how to test the accuracy of that model. And finally, looking at if the model provided, the study provided the source code. So an overview of all of this is shown here, showing that by and large, model validation has only occurred in about half of these studies over time, and that has not changed over time. The ODD protocol, since it was first introduced in 2006, has grown to be about 25% of the studies from year to year. Source code has increased over time, starting in the mid-2000s, and likely will continue to increase in the future as it becomes easier to share your models on online repositories like GitHub. In addition to my literature review, I also conducted field work at a number of sites. In 2014, I went to the Minnesota Zoo and took videos of the Japanese macaques. In 2015, I went to Ethiopia and observed the Hamadryas baboons. In 2016, I went to Gombe and observed the chimpanzees and olive baboons. And in 2018, I went to Ethiopia once again, but this time to observe gelatos. And so from all of this groundwork, I produced the model Beget. And as Mike has already mentioned, I really envisioned this model to be a generalized model for testing hypotheses in primate social ecology. And I had some more specific aims that I'll talk about now. One is that I wanted to test the performance of Beget, make sure that it was working properly, make sure that the virtual primates behave like their real life counterparts. Also, make sure that the plant distribution varies in response to the parameter settings that you can set with the model. This is very important because plant distribution is an important factor in primate socioecological models. And finally, to apply model validation to determine that Beget is working as expected. The next step was to test the necessity of the biological constraints that I mentioned previously. And finally, once I was fairly certain that Beget was operating as expected, to use Beget to test hypotheses. So some important hypotheses in this field, including the stability of breeding bonds on female group size, individuals should help others in proportion to their relatedness, and primates living groups to obtain better access to food resources. And I'm going to get into all of these in a little more detail, starting with talking about the virtual primates. So I opted to model, simulate 4 different primates, virtual primates based on the 4 primate species that I observed. So chimpanzees, gelatos, Hamadryas baboons, and olive baboons. And rather than going through some quantitative results that I have, I thought it would be much more fun if I showed you some videos of these virtual primates acting like their real life counterparts. So in all these videos, it's just kind of a slow motion view of what's going on in the simulation. On the right-hand side here, you have a feed for a lot of different things that are going on within these simulations, including that the females are shown in circles. There's a female there, males are in triangles. another one. Here we're looking at a homozygous male. In homozygous baboons, the males recruit females into their groups. And so here is a homozygous male recruiting a female group. And she's going to get that equal sign, and she's going to group. Then she's immediately going to mate with her. Chimpanzees fighting over a female. I'm going a little slow, so I might get past them, but you get kind of the idea. Here's a homozygous female giving birth. So in this group here, so we have the green group, which includes an adult male here, a female here, and then there's another female in this group. Oh, and she just gave birth to a little infant male. Here is a Delata unit in which this male is here. The mating opportunity for the female here. She just leaves that female. That's why we have a K there. She's putting down that female, and now she's ready to mate with her. I wanted to capture particular things about these different species, including their dispersal patterns. So in all of baboons, males disperse from the male community when they reach section 30. And so here is male. There's a male here that is about to leave the group. And when he leaves the group, so change colors and kind of move away from the group. If all of these are different, he eventually came to this group right here as he formed this group. So what is going on with all of these different behaviors that we're seeing? So at every given time step, for every individual in the simulation, the following occurs. One, that individual assesses their environment, which is drawn by the stacked line. This is what this blue male can see in his environment. Some plants, and then these two females right here. And then what he does with this information has to do with whatever is in his genotype. So depending on what is encoded there, he is going to make certain decisions. So one can think about these factors of the environment as being the keys that unlock particular genes in the genotype to access these different decisions. And so within each time step, a queue of decisions is formed based on what the male has decided to do at that time step. For example, here he's decided to mate with this red female. And so from that individual's perspective, you have this queue of decisions that he's going to do. He can only actually perform those decisions if he has enough energy to do so. And in this particular hypothetical scenario, he does. And so you can see the allocation of energy to these different actions through these type pieces here. And so these are different actions that he's performing at that time step. Here's what the genotype looks like. This is just a little snippet of the homozygous genotype. And so in each row here, what you're seeing is one of the genes, and it encodes some decisions that is made. So this particular one says, if I am an adult male and as an unrelated, unaffiliated living female, then I will expend some energy to recruit her into my group. These genotypes that these individuals have are heritable, they're passed on through reproduction, and they're also mutable. Some mutations can occur over generations. And these translate into decisions, as I mentioned. And the nice thing about these phenotypes is that you can export them from beget into text files and save them. And so users can save and import these files to create a lot of different possible scenarios. In addition to the genotype files, I also have population files, which save the starting state. Well, they are the state of every individual in the initial population. So when you import them into beget, that's the starting state of your population. So here is what the 4 virtual primates that I simulated look like in their initial starting state. And figures like that. These can also be saved externally as CSV files, in which the rows represent the individuals and the columns represent the attributes of those individuals. Moving on from the virtual primate to the plant distribution. So the way that I modeled the plant was heavily inspired by Conway's Game of Life, and the rules are shown here. And so how Conway's Game of Life works is that you start with a collection of cells shown here, in which some cells are alive, shown in black, and some cells are dead, shown in white. And then as the simulation goes forward, the rules dictate which cells come alive or become dead over time. So for example, a dead cell next to 3 living neighbors will become alive, whereas a living cell next to 6 living neighbors will die. This is just showing a stable state. This configuration is stable from time step to time step, as is this one. So as you can see, each one of these living cells is adjacent to 2 other cells, so they continue to live, and everything stays the same. Here's one that blinks up and down, and it can get more complicated from there. Here are a few gliders on the left here, showing that they retain their shape, but they glide across the screen. Here is a more complex game of life going on, and I was struck by how it almost looks like some variation in plant distribution going on. And I thought, what if we instead in Conway's Game of Life, we're just considering life as occurring with 2 or 3 neighbors. But what if we change the rules to something else, such as 2 to 6 neighbors or 0 to 8 neighbors? And so that's exactly what I did in beget. Is now you can set how many neighbors each of the plants prefer in that simulation. So here's just kind of a baseline 0 to 8, which shows a more homogeneous distribution of plants. 0 to 1, 0 to 3, 2 to 4. So we get a lot of different plant configurations depending on the preferences, the neighbor preferences of these plants, including some very patchy plant distributions here, which is ideal for modeling questions in primate social ecology. So to get into the simulations that I ran a little bit more, so overall for my dissertation, I ran a total of 4,608 simulations, which are visually shown here, where each one of these little squares represents one simulation. And I grouped them into these 16 groups here, which I call different worlds. So world A, B, C, and so on. World A is the baseline world, so those are the baseline parameters that I used. And then each one of these other worlds deviates in some way from that baseline. For example, world J is identical to world A, except that the interactions are aspatial, because I wanted to test whether or not it mattered if these primates lived in a spatial or aspatial world. And within each one of these worlds, I also divided the simulation and started each of these simulations with one of the 4 seed populations, the chimpanzees, geladas, hamadryas, baboons, and olive baboons. And then for each one of those collections of simulations, I varied those plant neighbor preferences. And so for each simulation, you have this unique combination of parameters. For example, this one is shown here, simulation D017B, which means that that simulation is from the world seed settings, the olive baboon seed population, and the plants prefer between 1 and 7 neighbors. And I did run 2 identical runs for each of those combination of parameters, and this is the second run B. And this proved to be a really nice way to encode what was going on within each simulation. For all of these simulations, I ran them for a total of 100,000 time steps. That was a big endeavor, and so unlike my previous simulations, I ran all of these at the Minnesota Supercomputing Institute, and even so, it took several months to do. And here's some results of this wide collection of simulations. So getting back to Conway's plants, if we're looking at the simulation A and looking at that plant distribution, here is what emerged from those simulations. So a really nice distribution of plants here. You can see these darker, more homogeneous plants in these simulation settings and down here. So that's indicating that darker color indicates that there's more energy available for the primates to eat. Whereas in this band, diagonal band in the middle, the plant energy is less abundant here. And then we also have this zone in which the plant distribution is very patchy. So moving on to model validation. So here's the modeling cycle, which is what we use to describe the developmental process of building a model. So we start with the target system. That's the real life system that we're trying to simulate with our model. We synthesize observations from this target system to create a conceptual model. And then we implement the microscale model according to that conceptual model design. The microscale model that we developed. And then we run systematic simulations and record the emergent properties, and that's what we get for our simulation output. And then ideally, what you have at that point is something that you can report the results of, and ideally that those results then say something meaningful about the target system. Likely that does not occur in the first time around the modeling cycle. And once you compare the simulation output back to the target system, you realize that some revision is required. And so you go through the development process again and again. Probably many, many times. I didn't count how many times I went through it, but probably quite a bit. But then the question is, when do you know to stop? When are you done? When are your results good enough? And that's where model validation comes into play. And that's what my colleagues and I did for a study that's in revision right now, is we based a model validation framework. We used a model validation framework and developed it further based on what Agaziak et al. Had done in 2014. And what we did was we took a simple hypothesis in which we hypothesized that in species with intergroup lethal aggression, these species are territorial, such as in chimpanzees, we would expect that in larger territory sizes, they would incur less risk from intergroup aggression. Whereas in smaller territory sizes, you would see a higher per capita mortality rate. And so I developed a model, which I call lethal geometry, which simulates agents who have these territories shown in these different colors here, and they're aggressive, and so they attack other individuals who are not in their groups. And from the simulation output, here's what we get. So we plotted the exact same information, territory size versus per capita mortality rate, and we found a similar inverse relationship. And to complete the cycle here, we also investigated what was happening in the target system and looked at some empirical data and plotted it with the same axes here and found the same inverse relationship. And the point of all of this is to point out, so this is kind of moving along the modeling cycles here. And this is kind of an ideal situation in which we can be fairly certain that lethal geometry is operating as we expect it to, because we have such strong consilience between all of the elements in the modeling cycle. And so that's what our model validation framework is all about. And so I applied this model validation framework to beget, and I can't go through all 10 model validation tests that I did, but I did want to highlight something that was visually appealing for you to see. So here I am loading a particular population and genotype file into beget. So here's what the interface of beget looks like. And so I'm kind of taking advantage of the fact that you can easily import and export population and genotype files. And so I edited particular population and genotype files such that these agents did very precise behaviors. So for example, in these simulations here, you see that the agents are either going around in a circle or going in a straight line here or there. And along the way, they're eating plants. And the end result of that looks like this. And that's what you want. So this is the validation test. If all the agents produce this pattern, then I can be reasonably assured that things are operating as expected. And there are more than just this one. I did 9 different populations, but it's important to make sure everything's running properly. Another way to test that things are running properly is just to use logic to think about, well, do the primates behave as expected? Here's kind of just an overview of all of the different simulations. So each one of these cells again represents one of these simulations, and they're grouped by their world, so world A, B, C, D, so on and so forth. And you can see that the black ones went extinct. And these match very well to that diagonal I was pointing out earlier, where there was lower plant abundance, or there was a harsher environment with a more patchyly distributed plant. And let me just point out briefly that some of these simulations down here did not finish in the allotted 4-day time window, simply because the population sizes got too large. Moving on to testing the necessity of biological constraints, I'm going to focus in on the aspatial versus spatial debate. And so here again is looking at an overview of these simulations. And this time we're looking at deaths by infanticide per simulated individual. And as you can see, there's a striking difference between the world-based simulations here and the rest of the simulation. And so it appears that in the aspatial world, which is what world J is, the individuals had equal access to all of the other individuals. That's what it means to be aspatial. And therefore, some of these individuals had access to infants and killed them on a more regular basis than is expected by this more the spatial simulation of here. And finally, I applied to get to test some hypotheses. So first of all, we tested the stability of breeding bonds. So here's from our first study using beget, which was published in 2019. And we were interested in looking at the evolution of stable breeding bonds. And so what we did is we evolved a bunch of different males over several generations, and then selected out genotypes from the selection and assigned them to either a category of rover, rover meaning that these males typically like to rove around, go from group to group, find as many fertile females as possible and mate with all of them, versus loyalists who tended to stay with one group, just mate with the females in that group and stay put. And so what we did is we released all of these different males using these different genotypes into a bunch of simulated worlds of various female group size. in kind of a tournament style where we saw, okay, who secured the most fraternities in these different female group sizes. And so what we found is that the loyalists, the ones who like to stay put, stay with their group, did very well in the very small female group sizes, smaller than 4. But once that female group size increased, now the rovers took over and they secured more of the fraternities. I also wanted to test Hamilton's rule that individuals should help others in proportion to their relatedness. And what I did for this is I started out each of the 4 primate populations with some genes that code for decisions to help each other. When individuals help each other, they actually increase the survival chance of the individual that they're helping. But I set those initial genes to be 0, meaning that they didn't actually perform those behaviors. They just had the genes available in their phenotype. And so what had to occur over time is mutations had to occur such that these helping behaviors emerged. And I was interested in seeing, well, did these individuals help their kin more than their non-kin? And so I tracked these individuals and their helping behavior based on whether they were helping individuals who are half related to them, a fourth related, eighth related, or not related at all. And then I tracked how much energy were individuals spending to help individuals from these different categories. And what I found was, well, it looks like in chimpanzees and homo bias here, we see that there is more help going on to kin than to non-kin, and same with olive males here. But gelatos, interestingly enough, show the opposite pattern where they tended to help non-kin more than kin. And finally, I wanted to investigate kind of a long-standing debate in primate socioecology that primates live in groups to obtain better access to food resources. So the idea here is that if you're living in a group and you're a larger group than some other groups out there, you probably can secure the access to food resources simply because you have more individuals there to monopolize that food resource. However, you don't want your group to be too large because then that's more mouths to feed and there's more interspecific feeding competition. And so ideally, the optimal group would be somewhere in the middle of that. And that's exactly what I found in simulating virtual primates. So here, what we're seeing here is I tracked females throughout the course of the simulation. So many females were born and died during the course of the simulation. And so I tracked their average group size, which is based on what their group size was at birth and at death, and then their reproductive success in their life. So how many offspring did they have? And what I found is that the mean female fitness is these medium-sized groups. And so to revisit my aims again, for the beget performance, I found that virtual primates display behaviors typical of real-life primates. I found that the plant distribution varied in a very systematic way. And beget withstood scrutiny from time validation tests. Thinking about the biological constraints in beget, spatial relationships do matter. And in using beget for hypothesis testing, I found that the stability of breeding bonds depended on female group size. Hamilton's rule emerged under some, but not all, simulation conditions. And females generally had the highest fitness in medium-sized groups. All right, I want to thank a lot of people, but for right now, I just want to thank my advisors, Michael Wilson and Clarence Lehman, my committee members, Mark Tappan and Karen McNulty, all of my lab mates, Julia Fisher and Dietmar Zinner for including me in their baboon symposium, which then led to the Journal of Human Evolution publication, for Larissa Swedell, Peter Fashing, and Mia Wen for hosting me at their field sites. All the students of Ortho-G9 who were my guinea pigs and used the early versions of beget and gave me very useful feedback. To my friends and family for their support, and to the University of Minnesota, and in particular, this department and also COGS for funding my travels. You can download the Get for yourself if you just go to this link right here. Actually, this is a link to my profile, so you can see all of the models that I created are available for download. And that's all I have for you. Any questions?
How were you able to get all of that into a only 500-page dissertation? You don't have to answer that.
You just saw it. Yeah.
If you don't have to go back to the Hamilton rule results, but I'm wondering if you have a guess as to what's going on with the gelatos and why they're different.
I've thought about this and I've looked, there's a lot of data I collected, so I haven't looked through all of it yet, but I'm not quite sure. Nothing, I've thought about a few things, but nothing really makes sense yet, so I don't know. Maybe you have some insight.
Well, I mean, it was interesting because even the chimpanzee females, I think they were also putting more energy into unrelated individuals, if I remember the figure correctly. So it just, I don't know, it's interesting. I'm not sure what would be going on with that.
Yeah. And for those simulations in particular, they ran for 100,000 time steps. That was about 8 or 9 generations worth of time. I would love to see these guys 100 or more generations down the line to really see what's going on.
So, but how many different starting points for that 100,000? How many simulations of those that were there? Independent starting point.
What do you mean by independent?
Well, so you can run one simulation for a long time to get a result, but you can also run one for a long time to get a result, and then start from a new starting point, a new initial condition, same parameters, new initial condition, then run it again, and then run it again. In other words, what's the replicability among runs on that? It could be that you've got a highly volatile situation now with highly chaotic results, and then you get different runs each time on it, different results.
Oh.
You might get different results. You might tend to get different results each time.
I mean, I found some clear patterns in the results, and I also did a replicate where I had the run A and run B. So that was the same initial starting parameters, but 2 different runs.
Hey, so John, I noticed you talked a little bit at the M3B and 3C about differences between species, and I was just wondering what your thoughts are on what sorts of interesting findings you might get by modeling 4 different species, and might that be worth expanding further in the future when you want to write 1,000 pages?
Sorry, what was the question?
Like what do you see as the main takeaway out of including the multiple species?
Well, I didn't want to just do one because who knows what would be going on if I just had one. I thought, well, 4, I didn't want to do any more than 4. 4 was a bit my limit. But having all 4 of them, then I could kind of start to see, okay, these ones are acting like this. You could kind of see the individual characteristics, well, not of the individuals, but of the different virtual primates, where these were acting kind of like this, and these were acting kind of like this. For example, I don't want to go all the way back to that slide, but in the slide where I show the aerial views of all of the simulations and the plant distribution, the Hamadryas baboons somehow were able to transform some of those typically patchy plant distributions into a homogeneous plant world. So that was kind of a unique thing that the Hamadryas baboons were capable of doing for some reason. So that's the sort of nuance that you get when you run multiple primate species.
You know, if there's online questions.
There are no questions in the chat.
Okay.
There's a lot of congratulations.
Yes, I have one more.
Sure.
So I know you might have plans for how to apply to get in the future. What sorts of other questions might be useful for you?
I have this curiosity in trying to figure out the grandmother hypothesis. hypothesis is one of the things that I would like to do. There's an ongoing debate between two different labs who use agent-based microscale models and with different debates on what's going on with the grandmother hypothesis. So I kind of want to weigh in on that. It's just one thing.
Were you able to account at all for like the multi-level structure in Hamadryas and the gelato populations?
I didn't. There was so much going on that I didn't really track the multi-level structure of these societies. But they're kind of present just in the way that the get is set up, where the individual groups are the breeding groups, and then those groups can exist within a larger population and associate with each other. And they do associate with each other, but I haven't specifically tracked in any systematic way what's going on there.
You add cultural inheritance factor into this?
Absolutely. So kind of also to answer Tony's question again, another direction I want to go with the get is to include language, communication, information transmission, and therefore cultural transmission. I think that would be absolutely fascinating.
All right, that will be the end of the public portion of the defense. Thank you again, Christy, and thanks everybody for coming. People saying to close session, maybe we take 5 minutes and